Hello, good morning. How are you? I hope you're all fine and you're all interested in taking this mock test, and also the you are preparing so good for your ensuing CBSE board examination. So first of all, I would like to congratulate you because you are entering into a very crucial phase of your life. So I wish you all well and a handsome mark in your board examination. Well, this particular talk is going to be about the importance of taking a CBSE mock papers. See, at the end of a session, I mean a learning session in a school or elsewhere, you are faced with one predicament. That is, how best or how far I have gone in preparing for a particular topic or particular concept. So that feedback you will get only by taking small tests. Yeah, it may be a slip test, or it may be a half an hour duration or 1 hour duration, or it may be a revision test like that. So first you should understand what is actually review of your preparation. So in that sense, this CBSE mock papers uh, make a very good sense. The first thing, mock papers simulate real exam conditions. First thing, you should just leave the exam fear, and as long as you are very clear in your concept, you will have the extra confidence to face anything in your life. So that confidence drives you. That is a driving force. Same way here, the mock paper simulate real examination condition. It improves the time management. That is very, very important. So a time management. See, you have a set of duration in which you are tested in learned topics. So the time, then it reduces the exam fear. So you just walk into your room as though you are entering into a hotel or you are entering into a pleasure area. So that leaves a lasting impression. Then identifies weak areas. You should you should always understand, you should maximize your effort, okay, and your output. For that, you should know what is your weakness and what is your strong points. The strong point has to be sustained and the weak point has to be eliminated. So once you accomplish this, yes, you are the real winner. And it improves the writing speed. That is very important. Many people are, very excellent in learning, but awfully bad when it comes in writing fast, and also diligent writing, and also it is impressive writing with all the clear concept. Just you have to put it across in the paper. Then it helps practice diagram presentation. Many people, they stumble on this particular aspect. So they are awfully bad in making diagram, except for the record notebook and other things. That is, they have little time to concentrate on this. Then it is assertion, reason, and case study. This particular aspect, it is predominantly present in Mark 2, Mark 3, and Mark 5. So in that, you have to be very careful. This assertion reasoning mostly occurs in the MCQ type, where you have recognition, recall, skill based one, and also understanding and application. And you have very many varieties of question. You have multiple statement question, and you have statement 1 and 2 and match the following, like that. So many questions are there, and this will help you in cracking NEET UG also handsomely. Then what are the golden rules? So golden rule says minimum 10 to full syllabus mock test you have to do. So this is very, very important. You have actually totally you have in the plus one, you have of course the 19 chapters. Here you have some 13 chapters. So these 13 chapters you have to be very clear, and you have a good strategy here for you. One is you have to master NCERT line by line, and also concentrate on the previous year question analysis. And of course, chapter wise mock test will be providing you, and full length mock test also is there for you. Then there is a error analysis. This area is a very important area, because unless you know where you err, you cannot proceed further. So this helps you immensely to then and there correct yourself, so that the next step, that error is not repeated again. Then presentation skills. You see, for 1 mark, no actual preparation is needed. I mean, preparation is in presentation wise. It is, they are given options. You have to select option diligently. You do that. For 2 mark and 3 mark and 5 mark, you have to know the keyword based points, and you have to underline the important terms, draw a neat label diagram wherever necessary, and maintain a neat framework in the answer book. Then leave space between the answers, so that the valuation is done in a pleasant manner. Then follow proper question numbering. See, many people, in their hurry to complete the question paper, they may sometimes skip the section number and also the question number, relevant question number. Then you should not overwrite, so that gives a clumsy picture of your presentation. Then common mistakes normally students do. They skip the NCERT lines when they are not quite sure of understanding certain line. You have to read in context, and also you have to make your understanding clear. See, once your learning is proper, your memory is also proper. Then your retention will be long lasting. And once you that is retain it properly, you can anytime you can recollect it, and you can explain the same. And ignore the diagram should be avoided. Then you have not practicing assertion reason, writing lengthy paragraph, don't do that. Let it be point wise, and it should be very relevant, and it should be very apt, and also it should be very catchy. Then you have to manage the time diligently, and you practice it. Then final success formula, you have to master the NCERT, and you know the blueprint. Mock paper practice, concept clarity should be there. Then presentation should be very excellent, and it is a continuing process. If you are interested in taking NEET UG, then the process still continues till you take the NEET UG examination. So this, if you follow this, the you will score the maximum mark in the CBSE biology paper. Yes, now let us get into the actual CBSE sample paper given by the board, and let us concentrate ourselves into section A, section B, section C. Yes, this is the section A. This is multiple choice question. So, in the male gamete, in plants are formed by, when it is a gametic formation, you all know it is gametogenesis. So, gametogenesis, 2n number becomes n. So, you have the, in the case of plants, you have microspore mother cell, and you have megaspore mother cell. And here microspore mother cell, one mother cell gives 4 daughter cells, while here one mother cell gives, even though it gives 4 daughter cell, the 3 are degenerated, one is alive. So here in this case, you have the cells like this. This is exine, and you have intine. You have what is called as a vegetative cell, and you have a generative cell. And here it should be borne in mind, the male gamete plants are formed by, very clear, it is the mitotic division of nucleus of the generative cell. So generative cell only forms 2 cells, and these 2 male gametes, they descend down in the pollen tube on the stigma tip. Then you have, so primary endosperm nucleus is called as pen. So you have here in the antipodal cell, and here the central cell, and here egg, and also it is flanked by synergid cells. So here you have a male gamete and a female gamete, a male gamete and 2 polar nuclei, a female gamete and 2 synergids. Here a male gamete and 2 polar nuclei. So this is what we call as triple fusion. So syngamy is egg plus the one male gamete which results in zygote, the other one is triple fusion. So this is what in later on converted as endosperm. Then you have during menstrual cycle of human female, formation of graphene follicles stimulated by secretion of which of the following gonadotropins. So FSH and LH, this is the answer. Follicle stimulating hormone, luteinizing hormone, they are very much needed. The luteinizing hormone surge during the 14th day of menstruation, that results in what is called as ovulation. Okay. Then you have the fourth question, the experimental proof of the thermal stability of genetic material was first provided by experiments. Yes. So Frederick Griffith, actually you know that there is a transformation principle, and they explained, the R type, S type. So rough type, smooth type, this is virulent and this is non-virulent. And in spite of heating and making the smooth cell, it a heat killed one, still the DNA molecule doesn't seem to lose all its character. So that is how the experimental proof on the thermal stability of genetic material was given by Frederick Griffith. So that you should understand. And then you have fifth one, short stretches of DNA used to identify the complementary sequence of the sample are called, they are called as probes. So why are they called probes? See, the probes they are called, see, as you all know, in the PCR technique, and you have ELISA techniques are there as molecular diagnosis. You see, normal urine analysis or sputum analysis or fecal matter analysis, blood analysis, they will give the onset of diseases only after the symptoms, after the incubation period of the pathogenicity is what you call as shown. So that is a bit late, and the treatment also is complicated. Whereas when in a suspected person for a cancer, these props are used. The props, they have known sequences, and they will not pair the complementarity with the mutated ones. So that way we can easily understand whether the person is likely to have a disease or not, like that. Now the next one is the select incorrect statement among the following. Here you have the Hardy Weinberg, as you all know, so this is the one. Darwin, even though he given the original species, he couldn't explain how the variations occur. Whereas the Stubbins, Dobzhansky, and many more people, they formulated, and now we have a very clear population genetic based explanation for the evolution to occur. So here, in a large and random mating population, the genes of dominant and alleles of dominant and recessive genes, they are equal to 1. So here, that is 4 options are given. That is, it indicates the evolutionary change. Hardy-Weinberg principle says that phenotype frequency in a population. So here constant, this is called a genetic equilibrium and all. The answer is the B. Okay. So indicates when frequency measured differs from the expected values. So here, this is the binomial expansion of P plus Q, and when frequency measured differs from expected values, the difference direction indicates the extent of evolutionary change. Hardy-Weinberg principle says that phenotype frequency population are stable and constant from generation to generation. The gene pool, total genes and their alleles in a population remains constant. This is called a genetic sum total is allelic frequency they have given. Here, one should understand the phenotype frequencies in a population are stable and are constant from the generation. This is the incorrect statement. It is not so. That is, all others are correct. Here, why it is incorrect? The phenotype frequency in a population are stable and are constant from generation to generation. Now, here the phenotype frequencies, they vary. Okay. Then albinism is known to be due to autosomal recessive mutation. The first child of a couple with a normal, with a what you call as normal one, with a normal one autosomal mutation, the first child of a couple with a normal skin pigmentation was an albino. Albino is without the color. Then what is the probability that their second child will also be an albino? Here you should understand autosomal recessive mutation, the recessive only in double dose that will express, in single dose that will not express. So here you have, if it is, that is capital A small a capital A small a, then you will have capital A capital A capital A small a capital A small a small a small a. So already they say one child is affected, so the remaining is only 25% is have a chance. Then you have in cricket species, cricket is something like a grasshopper. The sound produced by the rubbing the wings or less together play a crucial role in attracting mates. Any change in the morphology of cricket legs could potentially affect their ability to produce a sound. A mutant cricket had their thicker hind legs. What would you expect for this? The leg mutation will not lead to speciation, and you have will not lead to speciation if they diversify into new habit. The leg mutation will have little effect on other external features and therefore have the little effect on speciation. The leg mutation will have no effect on behavior and thus have the little effect. The leg mutation might lead to reproductive isolation and speciation due to an effect on their mating call. See here, the four different options are given. What their ability to produce sound, that is they are unable to produce a sound because of morphological changes have occurred. So in what way that is going to affect the cricket species? You see here, the leg mutation might lead to reproductive isolation and speciation due to an effect on the mating call. So this is very correct. See, you have studied what is called as, that is you have the industrial melanism in the Betson Bettleria in UK before and after industrialization, the black, I mean peppered moth and the grey moth. So they actually changed due to the hardship faced due to survival. Same way here, when it is not making a mating call, as a male frog croaks during a rainy season to call the female for mating, here, since in the absence of the phenotype, I mean the suitable phenotype for making a call, it will lose the ability. So it will have what you call as reproductive isolation, then and speciation. So it will change into different species, not from the original one. So plasmodium is a pathogen that causes malaria. Identify the correct sequence of transmission of the pathogen. So this is another interesting question. Here you find the, you see they have given some sequence. So pathogen as it enters. So you have Anopheles female mosquito bites this, and then first sight here, you know, when it bites a person in the finger or something like that, it enters, it first enters into liver cell, it enters as a sporozoite, that is correct. Okay, in the case of Entamoeba histolytica, it is tropozoite, like that. Then it infects the liver, then erythrocyte, that is, here you have signet ring change. Mutra, that is like a what you call as finger, that rings on the finger. And then it eats away the content of the RBC, it bursts and forms the micro and macro gametocyte, and eventually due to a bite of a mosquito, it enters into what you call as the salivary, that in the alimentary canal, and then here you find two digenic parasite. This one, in the case of human being, it is temporary parasite host, whereas in the case of mosquito, it is a permanent host. Okay, so in human being, it is asexual reproduction. In mosquito, it is sexual reproduction. So the answer is D. So here, which mRNA will be translated to a polypeptide chain? So here you should understand the, here you will find the, always you should know, that is AUG, that stands for the coding methionine, and also you have UA, UG and UGA. So these three are called as stop codons. So naturally you have to look for the augurs. Yes, you have OG, but they have given 8 amino acid. So for one amino acid you have 3 nitrogen bases. So based on that you have to look for that, the stop codon and what you call as the start codon. So this is start codon, this is stop codon. Then you have 1, 2, 3, 4, 5. Yes, This is correct. The B option is correct. Then you have, in order to isolate genetic material of a bacterium, the cell must be treated with. You have different method for the isolating different materials. For example, if it is a plant cell, then cellulase is the right one. If you want to digest RNA, you have to treat with the ribonuclease. If you want to digest the DNA, you have to treat with the DNase. So, likewise, here it is the material, that is, you have taken a bacteria. From the bacteria, you have to take the genetic material, the main chromosome. Then the bacterial cell will be lysed. Once you use lysozyme, then RNA will be removed by ribonuclease. This will, proteins and histone will be removed by proteases. Chilled ethanol will lead to what is called as spooling. That is, the chilled ethanol treatment with the separated or precipitated DNA will be taken out by spooling. So the correct answer is A. Then you have integrated pest management involves. So here, what is called as IPE? The IPE is IPM, that is integrated pest management. Pesticides are very costly and also expensive, and also that is pollution, what you call making. Then insecticides, they may indiscriminately kill the useful, what you call as insect and other pests also. So we have to use judiciously, means with a lot of care we have to take in choosing a particular insecticide. It should be very specific. It should not be non-specific one, so that along with the specific one, non-specific organism also may die. That will collapse the ecosystem or the balance in ecosystem. So the option 1 and 2 are the correct answer. You have to use biocontrol agents. For example, baculovirus, and you have nucleopolyhedrovirus, such things you can use. Okay, that's called biocontrol agent. The answer is C. Then we have the question 13 to 16 contain two statement, assertion and reason. Answer these questions. So this you know. The ability of the pistil to recognize the pollen is the result of continuous dialogue between the pollen and pistil. The electrical dialogue, this is totally wrong. So here you should understand, yes, when the pollen grain enters the stigma tip, yes, there is a dialogue whether it is compatible, whether it could be accepted. First, it reads there is a chemical interaction between these two, pollen and the pistil, and if it is belong to the same species, it is identified, I mean recognized and accepted and allowed to germinate. And if it is not of its own species, then it is rejected and not allowed to germinate. So it is not a electric dialogue, it is a chemical dialogue. It is a chemical light dialogue. This is something like a dialogue in the animal world, where you have fertilizing, anti-fertilizing in the sperm and the ovum interaction. So that's called chemotactic method. So same way here, it is a chemical reactions that take place between the pollen and pistil. So here the assertion is true, but R is reason is wrong. Then in this case, you have some organisms are better adapted to survive in an otherwise hostile environment. Adaptability is inherited and has a genetic basis. This is both A and R correct, and also the uh, explanation also correct. Then excess dose of coke or crack produces a sense of euphoria, increase the energy, and cause hallucination, and it interferes with the dopamine. So, in this case, yes, the excess dose of coke or crack produces a sense of euphoria. That is, in the drug addicted people, you can find this. They enter into ecstasy, and so that is called hallucination, and there is an increase in energy. That is correct. Then it interferes with the transport of dopamine. Dopamine is a material that is in the central nervous system, and that is blocked, so that you get this hallucination, which is very temporary. So both are correct. The option is A. So assertion, reason correct, explanation also correct. Then we find the Rosie was the first transgenic cow to make more nutritionally balanced milk for consumption by human babies. So that is correct. Okay. But the milk of Rosie cow contained human beta albumin. So alpha lactalbumin is correct. So A is correct, R is false. Well, so I hope you have understood what is this, and also in the subsequent videos we will discuss about the section B and section C, section D also. Well, before I close this talk on the uh, mock paper that is presented just now, let me introduce RGR Academy, that is headquartered at Ananagar. Here we have a sound faculty on which you can rely on, and we have a special residential program that caters to the need of NEET aspirant, NEET UG. Here our program is like this: we have a very small group, and this group starts work from dawn to dusk, from 6 a.m. to 10:30 p.m., and with a what you call as with a sufficient gaps for some sort of a rest. And here our program is tailor made for each and everyone's need, and we have a Paka mentorship, that is those who are trained by us, who joined in government medical colleges and elsewhere. They come on the weekend and they provide the mentorship to the aspiring NEET UG students. And another thing is, each group consists only of 20 students, and so it is one to one, and it is highly personalized um, what you call it as coaching program. And another thing is, here every topic, once it is completed, each topic is completed and the test is conducted, and it is fed to a AI generated analysis, and that based on the analysis report we find the strength and weaknesses of the student, and then we just eliminate those weakness area by just sitting with them and what you call it, clearing all their doubts, so that then and there this clarity is provided and the concept clarity is provided, so that the student get their extra confidence in facing the examination. And we have a very ambient environment here, and teachers are very friendly with the students, but yet they are highly disciplined, and there is a very good rapport with the parent, and now and then we what we call as enlighten the parents about the what's the progress, and also what need to be done on their part and on the faculty part, and frequent PTA meetings are conducted, and the grievances are redressed then and there. So it is a very very good opportunity thrown open to you. So it is up to you to make best use of us, and we are here to help you. And we all you are all welcome to join us for the ensuing crash course once you complete your board examination. And we do have online programs also, hybrid programs, and you just what you called us log on to our website, and also the numbers provided in our ads, and this video link is given here and there, and you can most you are always welcome to contact us, and we are here to help you a lot, and assuring you a good success in NEET UG. And this is the biology faculty saying goodbye to you. Thank you.